Dear colleagues, dear friends, welcome today for the second day of the Congress. This day will be dedicated to muscle disease mainly. We will have the first plenary session just now. That will be chaired by Volker Straub from Newcastle University. Other speaker will be Hans Jungblut from London and Rohit Agarwal from Pittsburgh. So, Volker, the floor is yours.
Yes, thank you very much, Gauthier, and thank you to all of you for coming to an early morning session. So this is the first scientific day of the conference. I'd really like to thank Gauthier Remisch, the organizing committee, for inviting me. It's always a bit tricky to chair your own talk, so I hope in terms of timekeeping I'll manage. Now, I was tasked to talk about an update on the limb-girdle muscular dystrophies, and I want to start with a disclosure statement. Now, I do have a lot of collaborations and partnerships with industry, but I actually don't think that there's any conflict of interest for my talk. Now, another disclosure is that I have to apologize. 40 minutes to talk about limb-girdle muscular dystrophies could be a bit boring for those who don't know about the limb-girdle muscular dystrophies. And it's a few years ago that I was giving a similar talk about the limb-girdle muscular dystrophies, and at that time I called it the ABC of LGMD. And the nomenclature, of course, was an alphabetic nomenclature, and you had the 1 for the dominant ones and the 2 for the recessive ones, and then the alphabet based on the time the limb-girdle muscular dystrophies were discovered. Now, the problem with the alphabet was that it ends with Z, and we actually had an LGMT D2Z. So there was definite agreement that we had to change something. And there was a consortium of colleagues who did this. I was one of them. And we said, well, with numbers, there is no limitation, so let's call it the 1, 2, 3 of LGMD. And then let's replace the dominant with a D and the recessive with an R. So the question is now, who actually knows these limb-girdle muscular dystrophies, really? Who knows what LGMD R16 is? And so I thought, well, maybe that would be a good topic actually for a plenary lecture where I have more than 30 minutes, because there are more than 30 LGMDs. And so, if I have about a minute per LGMD, we can go through all of them. And if you think, well, but it's pretty boring, you can at least then test yourself whether you know which one is the next one that's coming up. I might be surprised by some of my slides. And so I have about a minute per slide, and I'll introduce briefly the various limb-girdle muscular dystrophies, and I might not always use a full minute for each of them, and I might use a bit more for some others. So the structure of the talk will look like this, okay? So I will have the name of the limb-girdle muscular dystrophy on the top of the slide. The name should include the protein as well, but sometimes they are abbreviated, and I'll list them in full. And then I'll focus on the diagnosis, okay? And for the diagnosis, if available, I'll show you some patient features and also the range of presentation from childhood to adulthood, if they are also childhood onset LGMDs. I'll tell a bit about the gene or the protein name, a bit about the genetics, and if there are, for example, founder mutations. And I'll also look at the MRI. I'll only go into the lower legs because otherwise it would be too complicated. And I will also then go into some diagnostics, subcellular localization, see, and then are there any patient organization outcome measures? Where do we stand in terms of clinical trials, and what are the organ systems involved? And when my time is up, I get a warning. Now, because all of you will look at the little rainbow, I'll stop this for the next slides. It will also just irritate me. So let's start with the first one. LGMD-R1, or calpain related, is really the classical, the common, so classical common calpain limb-girdle muscular dystrophy. It wasn't the first one that was sequenced, but it deserves to be the first one to be named. And it shows onset in childhood often, but in adulthood. The protein is a calcium-activated neutral protease. It's a cysteine protease, and it's probably responsible for remodeling also the cytoskeleton after damage. If it's not there, damage gets worse. The MRI is very typical. It really shows pelvic girdle involvement and particularly hamstring muscle involvement, and in the lower legs, the soleus and the medial gastrocnemius are the first one to be damaged. You can see loss of calpain-3 on Western blots. Immunohistochemistry doesn't really work. It's a cytoplasmic protein. There are patient organizations as well, Cure Calpain-3. There's also one in Australia, the Daniel Ferguson LGMD Foundation. And there's a patient registry, which is very important. I think in terms of outcome measures, we will use a North Star ambulatory assessment for limb-girdle-like muscular dystrophies, maybe also the motor function scale, MFM. And there are currently natural history studies on its way, and fairly soon I would assume also clinical trials. In terms of the involvement of organ systems, you really have skeletal muscle involvement, including respiratory muscles, but no cardiac. There are animal models. Animal models are not that great. Now, the second one is LGMD-R2, or dysferlin related. And again, this is the second most common limb-girdle muscular dystrophy globally. It doesn't affect small kids. It just says it's mainly adults, actually. You will find that patients present in their late teenage years, but mainly in their 20s. The MRI, there are known founder mutations, as for calpain as well, many of them in Spain as well. And so you see some asymmetry. The femoral quadriceps muscle is involved, and in the lower legs as well. And we know that there's a phenotype that has been called Miyoshi myopathy. I think it's one and the same disease, but there is distal involvement. For immunohistochemistry, you can see loss of dysferlin. You can also see this on Western blots. It's important that the histology might show a lot of infiltration. Patients are misdiagnosed sometimes as myositis. In very rare cases, there's also amyloid. And it's a membrane protein also involved in the triad. Now, there is a very strong and supportive patient foundation, the Jane Foundation. They have a patient registry as well. There is a robust validated outcome measure, the North Star Ambulatory Assessment for dysferlinopathy, now also called North Star Ambulatory Assessment for limb-girdle-like muscular dystrophies. There are now natural history studies. There are animal models. Again, they're not necessarily great, but they're good knockout mice. And we are just about to publish standards of care. This took place 2 days ago in Brussels. This meeting, a final ICNMD supported workshop, also supported by the Jane Foundation. And the, again, clinic really focuses on skeletal muscle and on respiratory, but there's no cardiac involvement. The next one is alpha-sarcoglycan related. So the sarcoglycanopathies have 4 forms of limb-girdle muscular dystrophy. This is probably one of the more common ones, also depending on founder mutations. The phenotype is classical for limb-girdle muscular dystrophy. They have often calf hypertrophy, very prominent shoulder girdle involvement as well. And on MRI, you often see a discrepancy between involvement in the pelvic and thigh muscles with fairly well preserved leg muscles or lower leg muscles. The sarcoglycans can be detected on immunohistochemistry because they form a complex. If one of them is gone, often the others are affected as well. And you can also look at Western blot. They're parts of the dystrophin glycoprotein complex. Now, there's a specific patient organization, and I only focus on the specific ones. There are, of course, umbrella organizations as well. The LGMD2D Foundation. I would think that as outcome measures, we can also use the NSAD, probably also the North Star Ambulatory Assessment. And there have been pilot trials in gene therapy as well in Columbus, so something where we are still waiting now for randomized control trials, but it's an exciting development to see that there are now first interventional trials in the sarcoglycanopathies. Now, in terms of care and treatment, we also need to pay attention to the heart, even though in alpha-sarcoglycan it's very rare. Now, the second sarcoglycanopathy, beta-sarcoglycan, is actually a very rare sarcoglycanopathy. It's rarer than alpha and gamma. What's interesting in terms of the phenotype, there are overlaps with Emery-Dreifuss in terms of contractures, also with actually collagen 6 with rigid spines. The onset and the diagnostics for all of them are very similar. So the MRIs, you can't distinguish one from another. There's a specific patient organization as well for LGMD-R4 or beta-sarcoglycan. And again, in terms of outcome measures, where we stand in terms of clinical trials, it's similar to alpha-sarcoglycan. It's a bit more advanced here because there's a gene therapy trial from Sarepta underway. And the animal models are extremely good, particularly the mouse model for all of the sarcoglycanopathies. And again, we need to take into account that the heart is involved. Now, for gamma-sarcoglycan, which is the next one, R5, it's very similar. Again, phenotypically, you cannot really distinguish one sarcoglycanopathy necessarily from another one. As I said, the MRIs look very similar, undistinguishable from one another, always with a more proximal involvement with fairly well preserved distal muscles, immunohistochemistry or Western blotting. They're all part of one complex. Now, in terms of trial readiness, there is again a specific patient organization and a patient registry for the gamma-sarcoglycanopathies, the Kurt and Peter Foundation. And trials are on the way now as well. Good animal models for research. And here as well, we need to look at cardiac involvement in addition to skeletal muscle and respiratory muscle involvement. The delta-sarcoglycan related LGMD is a bit different. Clinically, it's also very prominent like a sarcoglycanopathy, but what's interesting is that there are a lot less patients, and this is throughout the world. So in large exome studies, this was really the rarest sarcoglycanopathy. Clinically, both on MRI and then also on muscle biopsies, very similar to the other ones. There's no specific patient organization, probably because it's so rare. There are actually currently no natural history studies. That was a mistake. I think what might be the case, and that's only speculative, that maybe in terms of the heart, there's a more severe problem. In the fish, it has been shown that delta-sarcoglycan is involved in cardiac development as well. So maybe they have a heart problem. This patient actually died because of cardiomyopathy, was on a transplant list. So they can have very prominent cardiac involvement. So the next one is R7 telethonin related. And as a clinician, I would say it is really a typical limb-girdle muscular dystrophy as well. They can have distal involvement. They can show, again, childhood onset to adults. The telethonin has also been the gene named TCAP as titin cap. It is located in the Z disc of the sarcomere, and it actually binds to the N-terminus of titin, so provides stability and elasticity as well to the sarcomere. You can see a loss of telethonin in immunohistochemistry, and also on Western blot. This was actually a patient with a partial deficiency. The MRI, I don't think that we really have enough data to define a pattern, but it clearly shows a limb-girdle muscular dystrophy. Interestingly, there are some distal muscles, including the TA, that are involved. Because it's so rare, there's no specific patient organization, and then I list often the Speak Foundation because it's an umbrella organization for limb-girdle muscular dystrophies. And with these ultra-rare diseases, of course, we don't have any validated outcome measures. There are animal models, flies and fish and mice for telethonin. And in terms of the clinic, it's interesting there's also an autosomal dominant hypertrophic cardiomyopathy, the type 25. So the heart can be involved, and if you have a patient, you need to check. I haven't mentioned this, all the recessive ones have actually high CKs, so these general things I won't list for each of them separately. Now, TRIM32, or R8, is also an interesting one because for many years there was one founder mutation in the Manitoba Hutterites in Canada, and it took about 15 years and an additional 7 cases were reported outside the Hutterites. So outside the founder mutation, it's extremely rare. TRIM32 is a tripartite motif-containing protein. It is this E3 ubiquitin ligase, so it's involved in the ubiquitin proteasome pathway for the degradation of proteins. What is interesting is that there is another name without that being known at the time. A sarcotubular myopathy is basically the same. It was first described actually with the same founder mutation as in the Hutterites. Because it's extremely rare outside the Hutterites, there's no specific patient foundation, and again, we don't have any validated outcome measures. And when I refer to RWD, this is real-world data, so basically, from publications and clinics, but without any systematic natural history study. In terms of the involvement of organ system, this is a pure skeletal muscle disease, and the 1 or 2 patients that I have seen had a clear limb-girdle muscular dystrophy. As I said, there aren't many patients described. Now, that's very different to LGMD R9 or FKRP related. This is actually in the north of England, where we have our clinic, the most common form, and this is due to a founder mutation coming from Scandinavia. And in Denmark and probably Norway and Sweden, this is the most common form of limb-girdle muscular dystrophy. Interestingly, the Manitoba Hutterites also have that same mutation, not just the one for TRIM32. So this is the fukutin-related protein. It's a Golgi resident glycosyl transferase, important for the glycosylation of dystroglycan within the dystrophin glycoprotein complex. The disease is outside that founder mutation, extremely rare. It's also extremely small. It only has 1 coding exon. The MRI has been studied quite extensively, and with almost all limb-girdle muscular dystrophies, we see that the sartorius and the gracilis muscles are always well preserved, whereas the hamstring muscles are affected. And what's also interesting, with all the dystroglycanopathies almost, the tibialis anterior is often a bit hypertrophic and well preserved. You can see deficiency of laminin alpha-2 on blots, but you can also look for dystroglycan glycosylation in these diseases. Now, there are specific patient foundations, the LGMD2I Research Fund and Cure LGMD. Both of them support a global FKP registry. In terms of outcome measures, there have been natural history studies, and there's been quantitative MRI, and we also looked at functional data, and really the FVC is the one thing that changes over a year. None of the other muscle assessments show any significant change in the population with the founder mutation. There are now randomized controlled trials, which is really exciting. There's also gene therapy starting very soon, and there are a number of animal models. Again, because this is a missense mutation, the founder mutation, it's more difficult. That animal model doesn't really show a phenotype. So, and the knockout mice show different phenotype, more congenital Walker-Warburg muscle-eye brain disease. So the brain can be involved in the earlier onset, but for most of the adult patients, it doesn't really matter. But the heart is really something that needs to be tested all the time. Then LGMD R10, titin related, and titin is of course a bit the chameleon within the muscle diseases. Now the initial limb girdle muscular dystrophy was described by Bjarne Ut in a large Finnish pedigree where there were patients with the distal, tibial myopathy that were heterozygous for the mutation, and then the homozygous ones had the limb girdle phenotype. As I say, it's quite a mixture and a broad pattern, and when do you call it a limb girdle muscular dystrophy is difficult to say. There are founder mutations. There are also, of course, everyone knows who's doing sequencing frequently, variants of unknown significance in the titin gene. I think it's a really important differential diagnosis. We don't know what your patient has, always think of titin. MRI patterns can be quite typical. I think the adductor magnus is always very well preserved. The semitendinosus is severely affected, and of course in the lower legs, the TA and the anterior compartment. Now, titin is difficult to do routinely on Western blots, but these patients often show a secondary reduction of calpain 3 or loss of calpain 3 as here. There's also often an accumulation and aggregation of myofibrillar myopathy or myofibrillar proteins like myotillin here. And in terms of patient organizations, there is a registry under the Congenital Muscular Dystrophy International Registry. There are no international natural history studies so far. Animal models are difficult. I think what is important, of course, with titin to always consider the autosomal dominant cardiomyopathy as well. Now, we are going into territory where people become less familiar with LGMDs. This is LGMD R11 or POMT1 related. Now, this was first described in congenital muscular dystrophies and muscle-eye brain disease and Walker-Warburg syndrome. But there is also a spectrum, and there are patients with limb girdle muscular dystrophy. So this is the protein O mannose transferase, which catalyzes the O mannosylation of proteins and is important for the mannosylation step, the first one of the mannosylation of dystroglycan. So therefore you can also see that in patients with POMT1 loss of function completely, this is a Walker-Warburg syndrome patient, there's no alpha-dystroglycan staining, whereas it's reduced in the limb girdle ones. You can also see this on immunohistochemistry. Again, I think for the MRI, the tibialis anterior is always very well preserved. It is an ultra-rare limb girdle muscular dystrophy. It's resident in the ER. And in terms of clinical trial readiness, we are not really there yet. There are no natural history studies, no defined outcome measures. You can register under the ICNMD. And I think what is important here is to keep in mind that these patients always actually have brain involvement. Interestingly, larger cohorts have shown that you don't necessarily see morphological changes on brain MRI in the LGMD cohort of POMT1. And the heart can also be affected. Patients often have a microcephaly, actually. They can also have a rigid spine and quite a lot of contractures. And they all have a low IQ. Now, anoctamin 5, R12, this is a really important one, I think particularly for the adult neurologists, because this is the one LGMD where you don't really have kids that present. And it's not large males and small females that are affected, but it's predominantly males that are affected. It's interesting, but clearly there's a gender difference. And I'm just showing you the legs because the legs are the important part that are affected, and patients will sometimes say, I feel as if I have two parts of my body. The lower body is the one that is affected, whereas actually I have normal strength in my upper body. That changes over time. You can always see that they miss the medial gastrocnemius. They don't miss it, but it's severely affected. The first muscle on MRI that is gone in anoctamin 5. Interestingly, what you can see in this MRI from one and the same patient, the gluteal muscles are the last ones almost in the proximal muscles to be affected. It's very different from all the other limb girdle muscular dystrophies. In dysferlin, you see this a bit as well. Whereas the thigh muscles are the ones that are affected. The sartorius and the gracilis are well preserved, and also the short head of the biceps femoris until the end. You don't really find anoctamin expressed on tissue sections very well. There are plots, this is from Bjarne Utz's group, where in the membrane fraction of the muscle, you can see a loss of anoctamin 5. Again, it's not necessarily routinely done. The protein was also called transmembrane protein 16E. It is a transmembrane protein involved in intracellular vesicle. And so anoctamin 5 actually suggests that it's an anion channel as well with 8 transmembrane domains. This is where the name comes from, anoctamin. But we don't know much about its channel function. Now, there is a specific patient organization as well, the LGMD 2L Foundation. I am convinced that we can use the NSAD as a robust outcome measure for this patient group as well. And the GRASP consortium in the US is now doing natural history studies as well. It's quite interesting that in the mice, it's also a male predominance in terms of severity and the ones that are affected. It's a primary skeletal muscle disease, really, and has no cardiac involvement. Now, Fukutine related, of course, we all know that there's Fukuyama congenital muscular dystrophy, which is a more severe phenotype than limb girdle, and the Fukuyama mutation is already a mild mutation, but there are even milder mutations that can cause a limb girdle phenotype. It is ultra rare as well. It's one of the dystroglycanopathies. This is why you can look at, dystroglycan staining with laminin overlays or the 286 antibody, also on immunohistochemistry or immunofluorescence. Again, as with the other dystroglycanopathies, it's the TA that is well preserved. Other than that, you have proximal involvement. It's a Golgi resident glycosyl transferase as well, Fukutin. And in terms of patient organizations, the CMD-IR would register patients with gene defects in Fukutin, but we haven't really done any outcome measures or natural history studies in the limb girdle phenotype. As with the other dystroglycanopathies, you have to also look at the brain and the heart in these patients. Now, POMT2 related is very similar to POMT1 because these two mannose transferases are both involved in the first mannose of dystroglycan. It's probably a bit more common, and I think this was a bit surprising when we did large exome studies to see that there were a number of undiagnosed limb girdle patients that had POMT2 mutations. The MRI, again, like with the other, shows more hamstring involvement with the sartorius and gracilis well preserved. Whenever you see this, it's a limb girdle muscular dystrophy normally. And you can also do the immunofluorescence or histochemistry looking at dystroglycan glycosylation. Like POMT1, it's an ER resident glycosyltransferase. Because there are not many patients yet, we don't have cohort for natural history studies. There are good animal models though, and like the other dystroglycanopathies, always consider brain and heart involvement. Now, POMC-GNT1 was always listed as limb-girdle muscular dystrophy, R15 actually, but when we went through the variant annotation through the ClinGen group for limb-girdle muscular dystrophies, we realized that the reported mutations were actually all polymorphisms. And then we looked at more than 10,000 exomes, and I asked colleagues who were involved in exome sequencing for limb-girdle muscular dystrophies, and there wasn't a single patient. Now, there might still be limb-girdle muscular dystrophies with POMC-GNT1, but there might not, and it might tell us something important about this protein. Then dystroglycan itself, so this is now the core protein in the dystrophin glycoprotein complex, has actually a few reports with patients with limb-girdle muscular dystrophy. So these are mild mutations because you need dystroglycan to be viable. It's an ultra-rare limb-girdle muscular dystrophy. You can have childhood onset and adult onset, and there was an amazing paper in the New England Journal of Medicine by Kevin Campbell, really looking also at the core protein because that's reduced or shows a shift. Now, in terms of trials and readiness, again, there's hardly anything. There are really a number of animal models. If you do a knockout mouse, it's early embryonic lethal, as you would expect. In terms of care and treatment, as for the other dystroglycanopathies, you have to consider the brain and the heart involvement. So then there is kind of an outlier among the limb-girdle muscular dystrophies caused by plectin. Plectin is known to cause epidermolysis bullosa. It's an intermediate filament linker, so it links the cytoskeleton. It's one actually of the biggest proteins that we have. And it was first described in patients with muscular dystrophy and epidermolysis bullosa. There's actually a founder mutation as well, probably in Turkey. Many mutations are affecting exon 1. But what's interesting is they don't always need to have epidermolysis bullosa. Actually, a few patients have now been described that also show congenital myasthenic syndromes. This is why these patients are listed, because they can present with ptosis without any epidermolysis. So the plectin gene shows a lot of polymorphisms if you do exon sequencing, so you have to be careful there. You can do immunohistochemistry and also Western blot for plectin, but overall, it's an ultra-rare limb-girdle muscular dystrophy, and because of this, there isn't really much in terms of outcome measures or natural history studies. There are good animal models, and you can also register in the CMD-IR patient registry. Now, the heart has been reported to be involved, but it's also more multi-system disorder. In this first Japanese paper, for example, that patients had alopecia as well, they had dental malformation, urethral strictures as well, so you probably need to do a thorough investigation for other organs involved. Then TRPC11 or R18. Now, this is the trafficking protein particle complex, subunit 11, and it's actually important to transport probably vesicles from the ER to the Golgi. It has been shown to also show reduced dystroglycan glycosylation, and it might be related that it affects other glycosyltransferases. It's not quite clear why. There aren't any good published MRI data, so there's no real pattern, but it seems that there's an increase in the STIR signal. There is a patient organization, I don't know how active it still is, but it's still on the internet. There are animal models, but there are no natural history studies. Now, interestingly, there are also patients that have been reported to show the limb-girdle muscular dystrophy, but they can also have hepatic steatosis. So some patients have even been reported with congenital cataract, so it's a more multi-system disorder, but can present clearly as a limb-girdle muscular dystrophy. GMPPB related R19, so this is GDP mannose pyrophosphorylase B. It's involved in the glycosylation of dystroglycan. This is why it's causing a limb-girdle muscular dystrophy. But it's not in the ER or in the Golgi, it's actually in the cytoplasm. What's interesting in terms of the diagnostics for this one is, on the one hand, there's a phenotype that also shows a congenital myasthenic syndrome. And so you can treat some of those patients with pyridostigmine, for example. There's also a shift in beta-dystroglycan that you can use diagnostically for this patient group. These are some congenital myasthenic syndrome patients as well, just to show you the spectrum, but you can have a clear limb-girdle muscular dystrophy, very similar to FKRP. There's no specific patient registry, but you can register in the CMD-IR, and we don't have yet any clinical trials. Again, because it's a dystroglycanopathy, think about the heart and the brain. ISPD-related are 20. Now, there are a few really great publications about this. It's a dystroglycanopathy. Again, the CDPL ribitol pyrophosphorylase A is actually the gene name that has changed now. It's ultra rare. You can do the same diagnostics looking at glycosylation of dystroglycan as for the other dystroglycanopathies. It's cytoplasmic SGMPPB. And in terms of the registry and trial readiness, very similar stage, mainly real-world data, good animal models. And in terms of care and treatment, make sure you check the heart. And in the younger patients only, I think you probably need to also consider the brain involvement, not in the classical LGMD phenotype. So R21, that's an interesting one. I think it's interesting because it was discovered quite late by Carmen Parada's group in Spain. Clearly, those patients show a limb-girdle muscular dystrophy. There's no question. You have, prominent shoulder girdle involvement. There's also atrophy of the upper arm. What is so interesting is that for this protein O glucosyltransferase, that the MRI looks very peculiar. The periphery here of the lateral vastus muscle is well preserved, whereas the center is replaced by fat. It's a mirror image of what we see in Bethlehem myopathy. There is a link to the dystroglycan glycosylation. POLGLUT1 is an ER-resident protein, but it's not quite clear what role it plays in terms of dystroglycan glycosylation. It's also very rare, I think probably underdiagnosed so far. There are now good animal models as well, and there can be heart involvement, but there's no involvement of the brain reported so far. And then we have introduced some diseases into the limb-girdle muscular dystrophies that aren't traditionally seen as limb-girdle muscular dystrophies, but actually they are presenting as limb-girdle muscular dystrophies. One of them is R22 that is related to collagen and recessive mutations. Now, you know that mutations in collagen have a broad phenotypic presentation from Ullrich congenital muscular dystrophy to Bethlehem myopathy, but there are also patients that don't have these typical, elbow contractures, rigid spine, skin changes, but they present like a limb-girdle muscular dystrophy, and it's important to consider this. The MRI, in opposite to the POLGLUT1 that I just showed you, there you have the center of the vastus lateralis here preserved, and the periphery is affected and replaced by fat. Now, it's not that easy routinely to actually diagnose these patients by immunohistochemistry or fluorescence or by Western blotting. You can do double labeling with Perlecan, and that might give you an idea that there's loss at the basement membrane, or you can also look into fibroblasts and look at the expression pattern, but it's not that easy. There is a specific registry that's global, a global registry for collagen 6. You can also register under the CMD-IR. There are first trials that have happened more in Ullrich congenital muscular dystrophy, not in this limb-girdle phenotype. Of course, one of the hints is also that there is skin involvement with follicular hyperkeratosis or keloid formation. And another disease that we felt needs to be on the radar if you see a patient with limb-girdle weakness and limb-girdle muscular dystrophy is lamina alpha 2, even though this is traditionally associated with a congenital muscular dystrophy. But there are clearly patients that present like a limb-girdle, and these can be kids, but also adults. I mean, we have diagnosed patients that are in their 50s, you know, a long history of limb-girdle weakness. Formerly, it was called Mericin. And interestingly, I think the MRI has some similarities to Bethlem. They're both extracellular matrix proteins. This is in the basement membrane. But you also see good preservation of the central part of the muscle, also here in the medial gastrocnemius. It helps to do a brain MRI because you would see white matter changes. And of course, you can do both Western blotting and also immunohistochemistry to look at a deficiency or partial deficiency of lamina alpha 2. For this specific limb-girdle phenotype, there are no natural history studies yet. And heart involvement is probably very rare. Patients can still develop epilepsy and show white matter changes, so the brain needs to be looked at. Then R24, POMGNT2, I mean, that's an extremely rare dystroglycanopathy with a limb-girdle phenotype. It's a protein O mannose beta-1,4-N acetylglucosamine transferase 2, one of the longest names for one of the proteins. There are, I think, worse ones. And in terms of the diagnosis, look at lamin overlay or glycosylation of dystroglycan. You can also see that there's a deficiency when you look at dystroglycan glycosylation on immunohistochemistry. Like POMT1 and 2, this is an ER-resident glycosyl transferase. Because of its rareness, again, we don't really have much trial readiness or natural history data. It's quite interesting that the zebrafish shows a Walker-Warburg syndrome. There's no fly yet that I'm aware of, but there's a mouse model as well. And like for the other dystroglycanopathies, we need to look at the brain and the heart. Now we come to two more. This is BVS related. This is the blood vessel epicardial substance, also known as a Popeye1 gene. So these are membrane-bound genes that are involved in binding cyclo-AMP and the potassium TREC1 channel. The function isn't quite clear, but they can cause clear limb-girdle muscular dystrophy. There are only a few families that have been published, so no trial readiness or clinical trials yet. And the heart for the Popeye genes can be involved. And so for POPDC3, which is also a Popeye gene, it's a Popeye domain containing protein 3. Fairly similar. Here we can also see that the patients show quite prominent calf hypertrophy, shoulder girdle weakness, and I understand that there's now another large family that supports that this is a limb-girdle muscular dystrophy. Again, it's so rare so far, and there are only a few patients reported in one publication that there's no clinical trial readiness yet, and the heart can be involved. This is the last of the recessive ones, and there aren't many dominant ones. So this is, see, this is the problem when I'm the chair. So this is the JAGET2 related, has only recently been described. I saw this kid which had a clear limb-girdle muscular dystrophy, severe contractures, and so it's something that was discovered through exome sequencing. Very rare. There's only, I think, one publication. JAG2 is involved in the Notch pathway, and there might be some involvement of the brain as well. So these were the recessive ones. In terms of the dominant ones, there aren't many, so you'll be relieved to hear that. There's DNA JB6. This is the most common one, really. And so this protein is involved also in, it's a co-chaperone, so it helps to avoid aggregation of proteins, and you can therefore see accumulation if it's missing of proteins, also rim vacuoles. And there is a patient organization specifically for the dominant LGMDs, and it's not clear. It seems to be a susceptibility factor for cardiomyopathies in animal models, but it's not reported in patients, and it's questionable whether the CNS is really involved. Now, the transportin 3 gene is an interesting one as well. It's a nuclear import receptor for splicing factors, and so this is extremely rare as well, but shows a clear limb-girdle phenotype. I mean, the PACE cavis also suggests that there might be some neurogenic involvement. The same patient organization, and it's a pure skeletal muscle disease. And then this is the longest name, I think, sorry, of, was I stopped now? I'm the chair. No, good. What is really interesting in this disease is that the patients show a prominent rectus femoris, whereas the rest of the vastus is really atrophic. It seems that there might also be an elevated hemidiaphragm, and distal muscles can be involved as well. For all of the dominant ones, I think that I've mentioned, there is also a similar fibromyopathy phenotype. Now, these patients can also show cataracts, which is important. I won't go into this with too much detail, but there are dominant forms of calpain. So these are milder patients, clearly with dominant mutations, more with truncal weakness as well. The MRI is milder than the recessive form. And we have something similar, of course, with collagen 6, where the patients are more adults with dominant mutations, very similar to Bethlehem myopathy. It's just that they don't show the classical Bethlehem phenotype, more limb-girdle phenotype. So similar to what I showed you for the recessive ones already. And you have to think about the skin again. So that was basically the summary of all the limb-girdle muscular dystrophies. And my take-home message, so this is a slide that you need to focus on now, is that the most limb-girdle, the most important ones are for LGMD, calpain 3, FKRP, and anoctamin 5. Now, the ones that you really need to remember is calpain 3 and dysferlin. FKRP and ANO5 are more in Northern Europe and North America. Titin, I think we still have to learn a lot. With sarcoglycans, I think we have diagnosed the prevalent population and now have to focus on the incident population, and they are quite rare. Altogether, the dystroglycanopathies are important to remember. And then from the dominant ones, the DNA JB6 is the most common one. The differential diagnosis is broad, of course. You have Becker muscular dystrophy on the one end of the spectrum, then you go into the Emery-Dreifuss muscular dystrophies, myofibrillar myopathies, even FSH patients with an atypical phenotype. So it is something that is challenging to even say, when do you really have a limb-girdle muscular dystrophy? There are more natural history studies now underway, in particularly through the GRASP consortium across the States and Europe. There are first interventional clinical trials, which is extremely exciting, considering that the first gene has been sequenced in 1994. And sorry, the bad news is don't get used to the nomenclature because we might change it again. Now, there's also an LGMD Awareness Day in September that is celebrated, and you can decorate limes. It's actually quite fun. And the good news is also that Heinz Jungblut won't use his entire 40 minutes. So thank you very much for your attention. I don't know if anyone still has enough energy to comment or ask any question. Yes, please.
Susan Ward, CTAP. I thought that was absolutely remarkable. Will your group be coming out with any recommendations of how one can pass a presenting patient into one of these genetic causes?
So, I mean, this is why I listed the patient registries, of course, that I think it's important that if you have a patient and there's a patient-specific registry, that the patient is registered. I think there's a plea for natural history studies. With some of the rarer diseases, we need to work together to collect enough patients, if that was your question. So for calpain and for dysferlin, anoctamin 5, there are now natural history studies underway.
I guess what I meant was, if I'm a physician and I have a patient who appears to have a limb-girdle phenotype, what's the most efficient way for me to figure out where that patient fits?
My advice would be you need to know where your local or national expert sits. In the UK, we are the National Referral Center for Limb-Girdle Muscular Dystrophy, so it's easy. They're referred to us, but I think in each country there will be differences in terms of who makes the diagnosis. For some of them, it's difficult, unless you have access to good genetic testing and then downstream confirmation, doing an MRI. I would ideally require all of that. Okay, so yeah, thank you very much for staying with me, and it's my pleasure now to introduce my colleague Heinz Jungblut. He's Professor for Pediatric Neurology at King's College in London, and for many years, of course, has shown an interest in the clinical description, but also in the pathogenesis of congenital myopathies. In the past few years, he has focused more also on autophagy mechanisms, and he'll give us an overview about advances in congenital myopathies. Heinz, and there's also chocolates for you, after the talk.
And Volker, where's the... oh yeah. Well, thank you very much, Volker, and thank you very much for the invitation. These are my disclosures, mainly related to advisory activities for companies developing therapies for congenital myopathies, in particular centronuclear and myotubular myopathy. And the concept of the congenital myopathies was firstly introduced in the 1950s and the 1960s when distinct histopathological entities were described following the introduction of new histochemical histological techniques into the study of diseased muscle. And the four major conditions identified were central core disease, multi-mini core disease, Nemelin myopathy, and centronuclear myopathy. And the field seemed very neat and tidy at the time, with these very distinct conditions being characterized. But as time went by, things have become much more complicated and much more messy, considering quite significant histopathological and also clinical overlap between these conditions. And the field has further and very markedly changed following the introduction of next-generation sequencing into clinical diagnostics, resulting in a large number of new conditions where the patient was essentially screened because of clinical features of a congenital myopathy, but not necessarily because of a very distinct histopathological pattern on muscle biopsy. And what I want to do in my talk is I want to give an overview over the congenital myopathies. I want to highlight certain new and interesting manifestations of a classical myopathy, and also described selected congenital myopathies, and in particular congenital myopathies recently described which have an overlap with other neuromuscular conditions. And I want to finish with an overview on therapy development in these conditions. And I just have to say that I initially assumed that my talk was supposed to be 25 minutes, so I did not cover some aspects as extensively as I would have wished, but I tried to cover them with some of the other slides. And in clinical terms, although many features of the congenital myopathies are fairly non-specific, there are certain features, in particular distal involvement, extraocular muscle involvement, the presence of exertional myalgia or cardiomyopathies, which may already point you in a particular direction of a specific histopathological or even a genetic diagnosis. And one aspect I want to highlight here already is that in contrast to many muscular dystrophies, the respiratory involvement in these conditions can be very disproportionate to the degree of limb girdle involvement. And this is something which has to be anticipated in the surveillance and management of these patients. And the congenital myopathies are markedly heterogeneous, often due to mutations in genes which are functionally associated. For example, most of the genes implicated in enamelin myopathy play an important role in thick and thin filament assembly and function. And I've already highlighted two of the most important genes implicated in the congenital myopathies, the skeletal muscle ryanodine receptor and titin, which is one of the novel causes which has emerged more recently and which I think is likely to play as important a role as the ryanodine receptor. And in contrast to the muscular dystrophy, the integrity of the muscle membrane is typically preserved in these conditions, and that is the reason why most of them do have a normal CK. But there are a number of intracellular, intramuscular processes which are affected, and these processes mainly concern intracellular membrane trafficking and vesicular trafficking. But most importantly, thick and thin filament biology, but most frequently the process referred to as excitation-contraction coupling. And excitation-contraction coupling describes the process whereby a neuronal impulse is translated into muscle contraction through the release of calcium from the sarcoplasmic reticulum stores and subsequent interaction between thick and thin filaments and muscle contraction, and the process is subsequently terminated by reuptake of calcium in the sarcoplasmic reticulum through specialized ATPase, the SERCAs. And there are two proteins which are crucial in that process, and one is the CAGNA1S receptor, which is a voltage-gated calcium channel which is localized on the transverse tubules. And the other one is the skeletal muscle ryanodine receptor, which is localized on the sarcoplasmic reticulum profiles and plays an important role in releasing the calcium stores from the sarcoplasmic reticulum. And mutations in the ryanodine receptor have emerged as one of the most common causes of congenital myopathies and associated with an extremely wide range of clinical manifestations. And on one hand, you have a large number of both dominantly and recessively inherited congenital myopathies. On the other hand, you have a range of manifestations. Some of them have emerged more recently, which are associated with the with the anesthesia complication, malignant hyperthermia. in otherwise normal individuals. And very simplistically speaking, in pathophysiological terms, the mutations associated with congenital myopathy phenotypes are often associated with a reduced release of calcium during muscle contraction, whereas the phenotypes associated with malignant hyperthermia and related conditions are often associated with an excessive calcium release because of the mutation. And the first congenital myopathy to be identified was central core disease in 1956, and the closely related condition multi-mini core disease in the early 1970s by Andrew Engel and colleagues. And although there has been a wide range of genes associated with course-on muscle biopsy, dominant and recessive mutations in the ryanodine receptor are by far the most common genetic causes of these conditions. And central core disease is a dominantly inherited ryanodine-related myopathy, often associated with marked variability within the same family. Occasionally, the parent of more severely affected children may have only very mild difficulties, for example, with sport, but no major weakness. One very consistent clinical feature in ryanodine-related central core disease is that these patients have a very distinct muscle MRI, and even if the histopathology is not as typical, the muscle MRI usually is very consistent and can actually help to establish the diagnosis in this condition, and also to distinguish ryanodine-related central core disease from core myopathies of other genetic causes. And central core disease is often associated on the physiological level with a leaky calcium channel which loses calcium at rest and therefore results in reduced calcium stores during weakness and subsequent weakness. And multi-mini core disease is most commonly caused by recessive mutations in the ryanodine receptor gene. And these patients are usually more severely affected. They often have more pronounced bulbar and respiratory involvement. But the most striking feature in these patients is the marked degree of extraocular muscle involvement, pronounced on upward gaze and on abduction. And that is a feature which distinguishes recessive forms of ryanodine-related myopathies from dominant forms such as central core disease, but also from other genetic causes of mini-core myopathy, such as recessive mutations in the selenoprotein N gene or selenon gene, where the extraocular muscles are typically spared. And I also wanted to highlight that although some of these patients may have a completely different histopathological phenotype, on the clinical level, ryanodine-related centronuclear myopathy or congenital fiber type disproportion look very similar to multi-mini core disease. So essentially, that is a spectrum of a very similar phenotype, not on the histopathological, but on the clinical level. And I think in one of my earlier slides, I did indicate that usually the CK levels are normal in the congenital myopathies, and that holds true for most conditions, but central core disease and other dominant ryanodine-related disorders may sometimes have elevated CK levels up to 1,000, 2,000. And the overlap with the congenital muscular dystrophy spectrum has been well recognized, but there has also been a recent recognition that some of these patients actually may mimic a limb girdle muscular dystrophy and not be immediately being diagnosed as a congenital myopathy, like the gentleman shown here in the picture. And in these cases, the muscle MRI may also be very helpful, because it is quite distinct from some of the muscle MRIs which Volker has shown earlier in the more common limb girdle muscular dystrophies, which have a different and distinct pattern from those who have ryanodine receptor mutations. And one very interesting aspect which has only emerged in recent years is the expanding spectrum of neuromuscular phenotypes associated with malignant hyperthermia, which is another manifestation of a mutated ryanodine receptor. And just to give you an idea in terms of numbers, malignant hyperthermia associated mutations are estimated with a frequency of 1 in 2,000. So that means in the United Kingdom alone, there are between 35 and 40,000 individuals which are likely to be malignant hyperthermia susceptible, which is quite a considerable number. And the connection between malignant hyperthermia and myopathies was already recognized by Michael Denborough, who was a very astute anesthetist from New Zealand, who described malignant hyperthermia for the first time, and at the same time recognized that some individuals who are malignant hyperthermia susceptible also may have a myopathy on muscle biopsy, in particular central core disease. And together with his colleague King, a pediatrician, he also described a dysmorphic syndrome with a myopathy called King-Denborough syndrome, which is closely associated with malignant hyperthermia, but which is still not fully understood in terms of the genetics, because these children often have a malignant hyperthermia mutation associated from an otherwise asymptomatic parent, suggesting that there are additional genetic modifiers in this presentation. In recent years, there has been a recognition of additional malignant hyperthermia associated phenotypes, and I just want to highlight 3 of them. The first one is the association with exertional rhabdomyolysis, and in recent years we have found that ryanodine mutations are a very common cause of rhabdomyolysis, typically triggered by exercise, but also occasionally by viral infection or, for example, by other recreational and medical drugs. And these individuals are not usually weak. On the contrary, some of them are actually very athletic and engaged in a wide range of different sports. However, what you do occasionally see is that some of these patients do have a mild ptosis or other subtle neuromuscular features. And what was very interesting in this original cohort is that although all the mutations we identified were known or putative malignant hyperthermia associated mutations, only one individual had a malignant hyperthermia reaction, suggesting that rhabdomyolysis may actually be a more common manifestation of malignant hyperthermia than malignant hyperthermia itself. And then we had this very tragic but also very interesting case of a 14-year-old boy who developed a fatal malignant hyperthermia reaction because of cerebellar swelling and subsequent coning when he was anesthetized for suspected appendicitis and developed a fulminant malignant hyperthermia reaction. And what was quite apparent was that in 48 hours before the triggering anesthesia, he had gone on a 20-mile charity walk, so had exercised intensely. And one observation in malignant hyperthermia, which has not been fully understood so far, is that many malignant hyperthermia susceptible individuals may have many anesthetics with triggering agents, but do not suffer a malignant hyperthermia reaction, whereas on other occasions they may develop a full-blown malignant hyperthermia episode. And because of the case history of this boy, we hypothesized that malignant hyperthermia may be due to a combination of a triggering anesthetic given on the background of a pre-damaged muscle. So we reviewed the malignant hyperthermia cases from 5 different malignant hyperthermia centers, and we identified 41 individuals who had exercised very intensely within 48 hours before the triggering anesthetic. And when I say intensely, I'm talking about activities such as cross-country skiing or ice hockey and other very intense activity. And when these individuals were anesthetized, often because of a fracture they suffered during these activities, they developed a fulminant malignant hyperthermia reaction. But we also compared the anesthesia with uneventful anesthetics in the same individuals. And we realized that they did not trigger and did not develop a malignant hyperthermia event when they had not exercised before the malignant hyperthermia. So that essentially does suggest that malignant hyperthermia is a compound event which reflects both a genetic background but also additional environmental factors such as exercise. I also wanted to highlight another manifestation of ryanodine mutations often associated with malignant hyperthermia, and that is the association with periodic paralysis. We had initially seen that in a single case some time ago, and it was uncertain if that was a coincidental observation or if there was a causal relationship. But we have subsequently identified a larger number of individuals where this was a prominent feature who suffered sometimes very profound episodes of periodic paralysis and who on a clinical assessment and a neurophysiological assessment also had additional features of periodic paralysis such as a positive McManus test. And the third association I want to mention is a non-neuromuscular association of ryanodine receptor mutations, and that is a mild bleeding abnormality we have seen in patients with central core disease, but also in patients with malignant hyperthermia. And this bleeding abnormality usually manifests in females with postpartum hemorrhage, menorrhagia, and in males with a milder phenotype characterized by nosebleeds. And the bleeding abnormality is due to smooth muscle involvement in ryanodine-related myopathies, also in vascular smooth muscles. And because of that smooth muscle involvement, the muscle does not contract properly, therefore you get prolonged bleeding. And there's also a suggestion that these patients do have a general smooth muscle phenotype. So for example, often they have unexplained bladder problems, unexplained bowel problems. But what is most interesting that both in the mouse model of malignant hyperthermia, but also in patients, the bleeding phenotype appears to be fully reversible with the ryanodine antagonist dantrolene, which is a feasible therapeutic option in this setting. And I think what has also emerged in recent years is that malignant hyperthermia is actually not a pure anesthesia complication, and that many of individuals who are malignant hyperthermia susceptible also have a mild myopathy. And I think there are a large number of patients seen in anesthesia departments with an expertise in malignant hyperthermia who actually also have very strong neuromuscular needs, which are currently unmet. And I think that is a very important population which in the coming years will need and warrant attention from neuromuscular myologists. And I think in my experience, these patients probably do not always end up in neuromuscular clinics. I see a lot of patients who were initially seen by rheumatologists, because they often have very marked pain phenotype, they have myalgia, and they are often suspected to have a rheumatological rather than a neuromuscular disorder. The next group of conditions I like to cover is myotubular or centronuclear myopathy. That again is a genetically very heterogeneous condition. Many of the genes, in particular myotubularin, dynamin 2, amphifusin 2, interact in pathways involved in intracellular membrane and vesicular trafficking. And they also have secondary effects on excitation, contraction, coupling, and other cellular pathways. And the condition I want to focus on here is X-linked myotubular myopathy, which is also one of the most severe congenital myopathies, characterized by profound weakness, bulbar involvement, and respiratory impairment, and also extraocular muscle involvement, which becomes manifest early in life. There are also milder forms in males, although they are rare. You may get very severe manifestations in females in the presence of additional chromosomal abnormalities or abnormalities of XN activation. There have been two observations in recent years, which I think are worth mentioning and highlighting. The first one, if you look on the picture here, on the bottom right is the wedding photograph of the oldest manifesting carrier of myotubular myopathy reported so far, and I think that is an emerging and important theme, that carriers of that condition actually do have a clear neuromuscular phenotype, and that is work led by my colleague Nicole Formans from Nijmegen, who described quite clearly that these females often have a very marked degree of weakness, facial weakness, but also in the limbs. And what is very interesting is that the weakness also can be highly asymmetrical, almost mimicking like an FSHD phenotype. And I think that is another group which probably will require more attention and input in the coming years. And another aspect I want to mention is the increasing realization that myotubular myopathy is probably not just a neuromuscular condition, but that it actually is a multi-system disorder, potentially affecting also other organ systems. And that is not entirely unexpected, looking at the ubiquitous expression and important biological roles of some of the proteins involved, in particular myotubularin and dynamin 2. And the multi-system aspect was already highlighted in very early studies, the study by Herman shown here in 1999, but has become more apparent in a number of larger natural history studies of myotubular myopathy, which have been published most recently. And the most important aspect of the multi-system involvement in myotubular myopathy is the hepatic involvement. And the most dramatic, but probably not the most common manifestation of the hepatic involvement is a sudden cystic transformation with often fatal bleeding called hepatic periosis, which is a very rare hepatic condition whose etiology in general is not very well understood. But I think what is actually even more common is a more general hepatic phenotype in these boys, and that involves general abnormalities of liver function, but also very commonly hyperbilirubinemia. And it appears that there also may be a specific defect how bilirubin is processed and transported in this condition. The fourth classical congenital myopathy, Nemelin myopathy, is also genetically extremely heterogeneous. At least the initially described genes all play an important role in thick and thin filament assembly and interaction. And the two most commonly involved are nebulin and mutations in the skeletal muscle alpha-actin gene. And Nemelin myopathy is a condition characterized by early onset, often very marked bulbar involvement, and often very disproportionate respiratory involvement, with patients going into respiratory failure even if they are still fully ambulant. And that is an aspect which has also been highlighted in the recent natural history study shown here, which did not only include nebulin mutated patients, but also patients with other genetic forms of Nemelin myopathy and unresolved cases of Nemelin myopathy. And you can also appreciate that the muscle MRI looks quite different to the muscle MRI seen in the rhinidine-related myopathies. There is a more distal involvement, particularly involving the tibialis anterior in the lower limb, and that also very much correlates to the clinical phenotype. And ACTA1 myopathy is also a clinically very variable condition. The most severe phenotypes are often associated with de novo dominant mutations in ACTA1. And I also wanted to highlight that recently there have been cases of an associated myopathy associated with different histopathological phenotypes, but also due to ACTA1 mutations. And I did mention the marked distal involvement in nebulin myopathy, but also other forms of Nemelin myopathy. And there's clearly an overlap with the distal autogryposis spectrum with regards to the genes implicated in Nemelin myopathy, and genes involved in Nemelin myopathy are also strongly represented in cohorts of fetuses presenting with the fetal akinesia sequence. So there is a continuum in both of these different spectrums. And I wanted to briefly cover the novel congenital myopathies, and I cannot cover each single one of them. I just wanted to highlight in an overview that many of the recently described genes also play an important role in intracellular calcium homeostasis, in excitation-contraction coupling, also in thick and thin filament interaction and assembly, but there are also a number of proteins which have been implicated in biological functions which have not previously been associated with congenital myopathies, and which clearly expand both the clinical, pathological, but also the genetic spectrum of these conditions. But one condition I wanted to specifically mention is the evolving spectrum of congenital myopathies due to mutations, to recessive mutations in the titin gene. And as Volker has already described, titin was initially implicated in limb girdle muscular dystrophies, distal myopathies described extensively by the Finnish colleagues. But most recently, it has also been recognized as a very common cause of congenital myopathies, described for the first time and very comprehensively in this seminal paper by Emily Oates and colleagues. And in histopathological terms, titin-related myopathies often have a very mixed histopathological picture, and that is the reason why they have not been as strongly associated with any of the classical phenotypes of congenital myopathies than some of the other conditions I've mentioned. And what you find prominently are central nuclei. Some of these cases have been classified as central nuclear myopathy. Cores are very common. Other cases have been reported as a core myopathy. But Nemelin rods, interestingly, although found in occasional cases as shown here, are not the most prominent feature. And on a clinical level, the phenotype is often very contracted, sometimes resembling Emery-Dreifuss muscular dystrophy. There's often marked respiratory involvement, but more prominently cardiac involvement. And the cardiac involvement can include both acquired cardiomyopathies later in life, but also quite frequently congenital heart defects. And I've encountered quite a number of patients who, because of a congenital heart defect and some dysmorphic features, were initially considered to have a non-neuromuscular genetic syndrome, for example, Noonan's syndrome. And I think that is a common theme in these conditions. I also wanted to emphasize, I've shown a very small picture of a family of a patient shown here. If you look after a patient with Titin myopathy, and particularly a pediatric patient with Titin myopathy, it is always or very often a condition of a whole family, because you also have to consider that the parents, the heterozygous carrier parents, may be at an increased risk of developing a cardiomyopathy, and in particular a dilated cardiomyopathy. So for example, in this family, the father died suddenly from a suspected alcoholic cardiomyopathy, but it is probably quite likely that the cardiomyopathy was a combination of lifestyle factors and also the genetic background. And one other thing I wanted to highlight, and that is work by my colleagues Martin Ries and Matthias Gautel, is that the interpretation of missense variant in Titin can be extremely challenging. Missense variants in Titin are present in around 3% of a normal population, so it is quite a challenge to identify how relevant they are actually to the neuromuscular phenotype. But applying certain biophysical approaches, in particular investigating the thermostability of this mutation, may hugely help in assessing their pathogenicity and their role in the phenotype observed. Yeah. Yeah. I'm much shorter than you, Volker. Okay, give me 3 minutes. I just wanted to briefly highlight that some of new myopathies are also implicated in excitation-contraction coupling, and that they have quite similar phenotypes. What they have quite in common is that all of them have malignant hyperthermia susceptibility, occasionally periodic paralysis, and quite similar neuromuscular phenotypes. So I go to the last bit of my talk, which is only a very few slides, and this concerns advances in therapy development in Titin-related congenital myopathies. And there are considerable challenges because many of these conditions are quite rare individually, and they also involve some of the largest genes and proteins, in particular titin and ryanodine, implicated in neuromuscular disease. And despite these challenges, there have been a substantial number of ongoing efforts, both in terms of basic studies, but also in terms of ongoing clinical trials. And I've highlighted here on the right currently ongoing clinical trials in green, and completed clinical trials in red, and one clinical trial in yellow, which is currently on hold. And because of the large size of the genes involved, so far only one condition, mild tubular myopathy, has been considered for viral gene transfer as a treatment. There are other treatment approaches. For example, the manipulation of proteins interacting in the same pathway, such as dynamin as an interactor of myotubularin and other proteins. And lastly, there are also approaches which try to modify the protein function. And that is something which is mainly being applied to the rhinodine receptor, which as a calcium channel can be subjected to pharmacological modification. And to end, I just wanted to highlight one example of a clinical trial approach in one of the severe congenital myopathies, X-linked myotubular myopathy, was based on very thorough animal studies suggesting that viral gene transfer in mouse and dog models are suitable to ameliorate the myotubular phenotype in these animal models. However, the trial is currently put on hold because of 2 deaths which are all related to hepatic complications. And I think that highlights that these conditions need to be viewed not only as neuromuscular conditions and, as I highlighted earlier, more as multi-system disorders with many aspects which have to be considered when treating the neuromuscular condition. I will finish here, and I want to thank a large number of collaborators. I also wanted to mention, because Volker also mentioned them in his talk, there is also a large number of patient organizations looking after these conditions, such as, for example, the Myotubular Trust, looking after patients with myotubular myopathy, and the RIA-1 Foundation, which is heavily involved in rhinodine-related myopathies. And lastly, I'd like to thank my team in London. Thank you.
So I don't have a microphone there, so thank you very much, Heinz, and you're right, I was the one who used up the time. Anyway, thank you very much for an excellent talk, and take your Belgian chocolate with you. Thank you very much. I'm really pleased to see that Rohit Agarwal is here, because when I got an email telling me that he's on his way from the States, landing this morning, I wasn't quite certain whether he'll make it, but your slides are up, so it's a pleasure to introduce Rohit Agarwal, who is a professor of medicine, medical director of the Arthritis and Autoimmunity Center in Pittsburgh, and a world expert in inflammatory muscle diseases. And so the floor is yours to tell us more about advances in this field of the neuromuscular diseases. Thank you.
Good morning. Thanks to the organizers for inviting me to present this talk. I know I have a very short time. I need to catch up. I will talk about clinical trial readiness in inflammatory myopathies, myositis. Here are my disclosures. So I have 3 learning objectives. I've divided the talk in 3 main parts. First, we'll talk about challenges in clinical trials in inflammatory myopathies. Obviously, any challenge that I pose here will not be as compared to the challenges that has been posed by previous speakers with ultra-rare diseases. Then we'll talk about classification and outcome measures in myositis. And lastly, we'll talk about lessons learned from the first successful randomized controlled trial in dermatomyositis, which is IVIG. So going into the talks, So first, just an introductory slide. I think this audience knows this very well. Idiopathic inflammatory myopathies, which in short we call as myositis, is a heterogeneous, rare, systemic autoimmune disease with primary target of muscle and or skin. It has various clinical subsets. Primarily, we used to call as adult polymyositis, but now we recognize that adult polymyositis is actually, a lot of those patients are necrotizing myopathies, some of them are anti-synthetase syndrome, and some of them may be even misdiagnosed as polymyositis are actually inclusion body myositis. So this true adult polymyositis subset has shrinked with the new 2 categories coming out causing necrotizing myopathy as well as anti-synthetase syndrome. Adult dermatomyositis is probably one of the largest subset, but we now also recognize clinically amyopathic dermatomyositis, specifically due to its association with severe interstitial lung disease and anti-MDF5 antibody. Then other subset, and then for this talk, when I refer to myositis, I'm only mostly referring to this clinical subset, where it's adult polymyositis and dermatomyositis subsets. Also juvenile myositis, which is mostly juvenile dermatomyositis, cancer-associated myositis, which is also mostly cancer-associated dermatomyositis, and some connective tissue diseases associated with myositis. For example, scleroderma or lupus may have an association with myositis. And last but not the least is inclusion body myositis, where I think is that we have the biggest challenges, but I will not talk about those in this talk. So first, FDA-approved therapies until 2021, we only had 2 FDA-approved therapies, prednisone and corticotropin, which were approved in 1952 around that time. And since then, we have had no FDA-approved therapy or any other major regulatory agencies in the world approved therapies until June 2021, when we got an approval. And so this poses a significant challenge. I mean, inflammatory myopathies, we treat these patients. These patients get better, and yet we don't have any approved therapy or any proven therapy for that matter. So when you look at the overall clinical trial scenarios through clinicaltrial.gov, for just specifically for dermatomyositis, you see that we are doing better in last 10 years as compared to the previous years. Before 2012, there were about 12 clinical trials. But since then, in last 10 years, there are 30 new clinical trials in dermatomyositis alone. And most of these trials, about almost 20 or 22 of these trials, are actually in last 5 years, 5 to 6 years. So we are definitely doing better in the realm of doing clinical trials and proving targeting different novel therapeutics, and then proving and then getting them approved. But if I compare that to the most successful disease in my world of rheumatology, which is rheumatoid arthritis, where there's a whole laundry list of drugs approved, biological and non-biologic drugs approved for its treatment, we see that partly is because they are doing a lot of clinical trials. Now, obviously, rheumatoid arthritis is a big number of patients, and it's not a rare disease by any matter. But still, they're partly successful because various parts of the world did a lot of clinical trials, tried a lot of therapeutic agent, and were successful. So I think if I compare, we are doing better, but if I compare to other rheumatological diseases at least, I think we are doing too little, and I think we need to do more of these clinical trials. Now, to aid that, we have a big problem here because we don't truly understand the myositis pathogenesis very well. It's complex, it's heterogeneous, and pathogenesis, honestly, is different for different subsets of myositis. But we do have some understanding now about key immunological players. So we may not know how these immunological players come together to cause myositis, but we know that what are some key immunological players. So what we can do is we can target some of these key immunological players one by one through different pilot studies, phase 2 trials, and phase 3 trials to find some good therapeutic targets and interventions for our patients. So now going into some of the challenges. So when we get to the clinical trial, what are the challenges are we facing in day-to-day clinical trial life? Well, the biggest challenge, I think, it's a rare disease. Now, it's not an ultra-rare disease, so it's not as difficult, but it's definitely a rare disease. So recruitment is the biggest challenge that we face when we do clinical trial. And then with the recruitment, obviously, cost and operations become difficult. Now, we did this rituximab in myositis clinical trial, which is still the largest clinical trial in myositis with 200 myositis patients with adult dermatomyositis, adult polymyositis, and juvenile dermatomyositis. But it took us 8 years and $8 million to get a graph like this. This is a shame in the sense, we need to do better, faster, and more meaningful clinical trials so that we can study different therapeutic targets a little faster so that we can eventually get our patients some treatments that are definitely possible. Another challenge is myositis is not a single disease. It's a highly heterogeneous disease, group of diseases with various different clinical subsets. So if you look at adult polymyositis or necrotizing myopathy or antisynthetase syndrome, it's very different from adult dermatomyositis and other diseases. Obviously, inclusion body myositis is a completely different animal. Also, when we get to a single myositis disease, let's say dermatomyositis, even then it's not a single organ disease. We have different manifestation, muscle weakness, rashes, interstitial lung disease. So in a clinical trial, you sometimes have to decide what is your target organ. Are you trying to see if the patient improves their muscle weakness? Are you trying to see if they improve their skin rashes? Or their interstitial lung disease. So even if we get to a single disease, it gets complicated. Also, fortunately, in last 2 decades, we have discovered many myositis-specific antibodies. Now, each myositis-specific antibody represents a unique clinical phenotype and pathogenesis. So one of the thought processes, why don't we target a serologically driven or autoantibody-driven subset? Well, in theory, that's better because you have more homogeneous patient population, more homogeneous phenotype, and even the pathogenesis is going to be more discrete. But the problem is then you're dividing a rare disease into a rarer subset, which increases the complication of doing a clinical trial. But the bigger problem, I think, is the lack of standardization of some of these myositis-specific antibodies. Big centers like us have no problems in getting these antibodies, but when you get to community practices and other areas, there is significant false positivity and false negativity for these myositis-specific antibodies. And also, when you look at the pathology, obviously I don't have to tell you this, but all of the clinical subsets of myositis are very different diseases. So we have another unique aspect or difficulty in clinical trials in myositis. So myositis is not a rheumatology disease and not a neurology disease, not a dermatology disease. It's been shared by various different subspecialties. And when that happens, each one of us, rheumatologists, neurologists, dermatologists, have different perspective, different experience of seeing these patients differently. So that definitely also leads to the complexity in terms of enrolling patients, in terms of doing outcome measures. For example, a neurologist may not be as apt as a dermatologist to do skin evaluation, and a dermatologist may not be as apt as neurologist in doing muscle evaluation. So that increases the complexity. But in rheumatology, we have seen much more complex diseases like this. Vasculitis is definitely far more complicated, far more complex, rare as myositis. But they have been very successful. They have 3 approved therapies so far with multiple clinical trials. It was only possible because they got together as a group of consortium with multiple collaborators around the world and started targeting different novel therapeutics. And finally, they have now many successful clinical trials and actually leading the way of how the rare disease can be tackled. So now going into some of the advances we have made in last 2 decades in clinical trials in myositis, some of the classification and outcome measures issues. So first of all, I want to thank Dr. Ingrid Lundberg from Sweden because she came up with a big group of neurologists, rheumatologists, dermatologists to develop this 2017 EULAR and ACR approved classification criteria for adult and juvenile idiopathic inflammatory myopathies. So if you see, they took about 41 centers in 21 countries with thousands of cases and controls with many, many variable studies and then validated in 2 independent cohorts to develop this classification criteria. Now, this classification criteria is sort of an upgrade from a Bohan and Peter classification criteria of dermatomyositis and polymyositis. Well, the Bohan and Peter classification criteria works very well for dermatomyositis because of specificity of the skin rash. But it doesn't work for polymyositis. It leads to misclassification and overdiagnosis of polymyositis. And secondly, it did not have an autoantibody, and there were other problems with Bohan and Peter classification. It was highly sensitive but not specific when you come to non-dermatomyositis entities or clinical subsets. The way this classification criteria works is similar to Bohan and Peter, where you have a lot of clinical manifestation. For each clinical manifestation, you get a risk score with biopsy or a risk score without biopsy, depends if you have a biopsy or not. At the end of the day, you add up all the risk scores to come up with a total risk score, and if the total risk score is above a certain threshold, you get a probable idiopathic inflammatory myopathy, and if it's above certain other threshold, then you get definite idiopathic inflammatory myopathy. Then finally, once you have met those cutoff, then you'd go by these classification tree to decide whether you're dealing with a PM subtype, a DM subtype, or IBM and other subtypes. The issue with this classification criteria, it does not represent the amyopathic dermatomyositis very well. It is not that useful for necrotizing myopathy, and also it lacks other myositis-specific antibodies. If you see, it has a myositis-specific antibody, which is great, but it only has Jo1 as a myositis-specific antibody. And as you know, there are many more myositis-specific antibodies. So, and also it really, the classification criteria does make you consider IBM, but this is not really made for IBM. This is made for non-IBM myositis. In terms of outcome measures, the IMAX group, which is International Myositis Assessment and Clinical Studies group, has done a tremendous work since early 2000 to develop outcome measures in myositis. And these outcome measures are listed here. So we have 6 myositis core set measures. What do you mean by core set measure? What it means is in any clinical trial and studies, we should be measuring these and other supplementary or exploratory core set measures. Now, what are these? These are physician global disease activity, which basically physician says on a scale of 0 to 10 VAS scale, how active is this patient disease activity is. Then patient global disease activity, which is a similar outcome for patients, muscle strength testing on MMT, physical function or disability through health assessment questionnaire, which is a patient reported outcome, lab testing with muscle enzyme, and extramuscular global, which means everything other than the muscle, tell me on a scale of 0 to 10. So as you can see, it's highly subjective, but it works. And then these are 6 different core set measures. So how do we combine them to give you a final score so that you can say overall this patient is improved in a clinical trial or not improved? And that's what led to this 2016 ACR-EULAR myositis response criteria. The way this criteria works, you have the 6 same core set measures, and based on the degree of improvement in each core set measures, and each core set measure is weighted, so based on the degree of improvement in each core set measure, you get a certain improvement score. And then you add up all the improvement score achieved in 6 core set measure to get a total improvement score. So, and then once you get a total improvement score, it's on a scale of 0 to 100. If you have higher the score, higher the magnitude of improvement, because that means more core set measures are improving with higher degree. And then you have 3 categories, minimal, moderate, and major. If you're more than 20 points on TIS, or total improvement score, you've achieved minimal improvement. If you have more than 40 points, you've achieved moderate improvement. If you're more than 60 points, you've achieved major improvement. Now, this was a very significant improvement from previous outcome measures that we have. This was a data and consensus driven. It's weighted based on all different values of different core set measures. It has absolute percentage change as a change, whereas relative percentage change is faulty. And also you can have a mean score of total improvements, or you can have a categorical minimal, moderate, or major improvement. And it's been approved by ACR and EULAR. But regardless, even though we have now ACR-EULAR approved consensus and data-driven myositis response criteria, we still have a lot of challenges. Most of these challenges comes from the 6 core set measures. Well, once we started with the 6 core set measures, there are many problems with these 6 core set measures that I would like to highlight. The biggest problem is subjectivity. Most of these 6 core set measures are highly subjective. They require training. They require expertise and experience, and different experience gives you different results, like a dermatologist versus neurologist versus rheumatologist. So, and that leads to decreased inter-rater reliability. So we have, even though we have made significant progress, we have still a lot of work to do. If you go to patient global assessment, oftentimes patient is assessing not the disease activity, but the damage, the comorbidity, or the adverse event influence the patient's rating of disease activity. Just by saying rate on a scale of 0 to 10 doesn't mean that the patient have all the understanding that is very complex to differentiate disease activity from damage. Even I cannot do in most of my clinics that. MMT, manual muscle testing, is also operator dependent. It depends on how the patient's day is going. If the patient has had a bad day, it might affect their muscle weakness that day. Same as for the examiner. It has a significant ceiling effect and is not very sensitive to change. Health assessment questionnaire has a ceiling effect problem. Muscle enzyme, the only truly objective outcome measure actually is normal in 20-25 dermatomyositis patient, as well as in some of the juvenile dermatomyositis patient. So it's not a great biomarker for dermatomyositis. It's great for PM and NM, but not so much for dermatomyositis. And then extramuscular has the same issue. How do you combine all the different organs in a single score of 0 to 10? But it works, and we know it works also. These kind of complex subjective plus objective core set measure put together works. We know that it works also from rheumatoid arthritis world, where they also have similar patient global assessment, physician global assessment, pain VAS, health assessment questionnaire, and an objective measure of CRP or ESR. We know this works because rheumatoid arthritis has had 10, 20 biological and non-biological drugs approved. So we know it works, but we need to do better. And in terms of doing better, I think if we bring objectivity in our outcome measures, that's going to be a significant advancement, given the complexity I've shown you with different doctors, different fields, managing myositis. So we have been studying different objective measures. So some of them are physical activity monitor, looking into PROs, handheld dynamometry, functional tests like time up and go and sit to stand, as well as objective skin assessment. So I'll just present the data on few of them that we have looked into in last few years. So first of all, physical activity monitor. To me, the idea of physical activity monitor is great. It's present. I think many of you might be wearing it right now. It can wirelessly transmit the data. It's low cost. You can get continuous real-time data. It's a very low patient burden. Patient doesn't have to do anything except for wearing. And it really doesn't have a cognitive aspect of data collection. And when we look into this, we looked into two groups independently looked into physical activity monitor. One is our group from Pittsburgh and another group from Paris, France, looked into it and showed that it was reliable, valid, and responsive in idiopathic inflammatory myopathies. So we need to introduce some of these in our newer clinical trials. Next is muscle strength testing is really key for myositis, but MMT is subjective, semi-quantitative. You can't expect a dermatologist to do as good as a MMT as a neurologist would do. So there are challenges there. So we looked into handheld dynamometry, which has been used extensively in the neurology world, as well as inclusion body myositis, but not really looked into as a tool for idiopathic inflammatory myopathy. So we looked into two proximal muscles, just the iliopsoas and the deltoid, and see what we get. And turns out it was highly reliable. Now think about this. I trained myself on YouTube and talking to a few neurologists. Then I trained another colleague by my training just in a matter of day. And with that kind of training, you get that kind of reliability is excellent. We had a very good validity as well as responsiveness. And then also patient-reported outcome measure. We need to do better work in PROs. Obviously, they provide a unique perspective of treatment effectiveness that we can't, as a doctor, tell. Patients can tell better. And in that realm, we have this, and also FDA and other regulatory agencies are very interested to, so much so, they have declared that a good PRO can be treated as a primary outcome measure in a clinical trial. And then this, I don't know how many of you may know PROMIS, but PROMIS is a patient-reported outcome measurement information system. This is a large US-based NIH initiative where they develop many state-of-art self-reported health outcomes. The good part is these are extensively tested, validated, and are reliable tools. They're made for low literacy population. They're validated in multiple disease states, multiple languages. They're relevant. Most of them are relevant to rheumatic and neuromuscular disease. And it's very time efficient. And they can also be given through computer adaptive testing, which decreases the time and effort even more. And we looked at in such one of the PROMIS measure for physical function. So PROMIS physical function, when we studied it, was highly reliable, it was valid, it was very responsive. In fact, it had a very high effect size in SRM, very low time to do and low burden, and very low ceiling effect and floor effects. Last but not the The last part of my talk is I'll talk about the randomized phase 3 randomized controlled trial, IVIG in dermatomyositis, and the lesson learned from it. It's called the PRODERM study. So the study was efficacy and safety of IVIG, which is octagam 10% in patients with active dermatomyositis. This was a randomized double-blind placebo phase 3 study. It was meant to be a regulatory clinical trial, a pivotal clinical trial. So first, the design of this study, we screened and randomized 1:1 half the patient into IVIG group at 2 grams per kilogram every 4 weeks, and half the patient into placebo group, again the similar dosing and infusion regimen. And then if at 8 weeks or more they meet the deterioration or they're worsening, then they can be moved to another group. But at 16 weeks was a primary endpoint, which was the total improvement score, which I've described recently. And then at 16 week, everyone would move into a phase 2 of this, not the phase 2, this next phase of the study, which was an extension phase where everybody will get 2 grams per kilogram every 4 weeks of IVIG for next 28 weeks, except if you're doing well at week 28, the dose could be reduced to 1 gram per kilogram every 4 weeks by the physician. The inclusion criteria was adult dermatomyositis as per Bohan and Peter criteria. As we've already discussed, Bohan and Peter criteria works very well for dermatomyositis. It doesn't work for non-DM inflammatory myopathies. And also at that time, we did not have the luxury of this new classification criteria. Each of the cases were examined for active disease by an expert committee. And also these patients were refractory, which means they have either failed steroid or other non-steroid immunosuppressive drug, or currently on those drugs but still have significant active disease. And last but not the least, they all had to have muscle weakness. And then primary endpoint was at week 16, which was the criteria I just described to you. 2016 ACR-ULR myositis response criteria. Again, it's based on total improvement score, which itself is based on improvement in 6 scores at measure, and 3 thresholds of minimal, moderate, and major improvement. Okay, so the results, all patients had symmetric proximal muscle weakness and has had typical skin rashes. Most of the patients met definite Bohan and Peter classification criteria. We enrolled 95 patients with 53 years of age as a median age, 75% female and 92% Caucasians. We screened 126 patients and randomized and treated 47 to IVIG and 48 to placebo, and 95% patients completed the first phase of the study, which was the randomized controlled trial, and subsequently extension phase was completed by another 72% patients. Now, the primary endpoint was this, the minimal improvement. How many patients or the proportion of responders who achieve minimal improvement at week 16. Now, IVIG group, 78.7% patients improved as compared to placebo of 43.7, which was almost the half the IVIG group. This was a highly statistically significant result with a delta of 35%. Now, that was the primary endpoint. But to me, I think it's a low bar because minimal improvement just gets patient a little bit better. So I really wanted to see what's the moderate improvement. And to my surprise, I was very pleasantly surprised that my moderate improvement was achieved by 68% patient, which is 3 times as much as an improvement that got in the placebo effect. And then the major improvement, which is very difficult to achieve, especially in week 16, was achieved in almost 32% patient, which is 4 times as compared to the placebo. So these were highly statistically significant results, no matter how you see it. And if you look at the total improvement score, or the mean total improvement score, where the treatment group or IVIG is represented by these blue-green dots, and the placebo is represented by the gray, you see the very early on there's a separation, and it continues to separate out all the way up to 16 weeks. When patients in the placebo group get IVIG, they also improve with similar slope and frequency and intensity. So to me, the lesson from this was that the outcome criteria worked, the total improvement score and this new ACR-ULR classification criteria worked, because I already knew IVIG works. So if the IVIG did not work in dermatomyositis, that would have been a failure of an outcome measure. So for me, actually, it was more of a success of an outcome measure than a success of the drug itself. Now, people would ask, you know, if the total improvement score improved, how do we know what component or what aspect of the patient improved? Did their muscle weakness improve? Did their physical function improve? Did their rash improve? So when we looked into that, almost everything improved. Their health assessment questionnaire, their skin disease activity, which is CDASI total activity, their MMT, patient global, physician global, and extramuscular, almost everything improved except for muscle enzyme, which we already know that muscle enzyme is a poor biomarker for dermatomyositis. Now, going into the adverse event quickly, there were significant adverse events, including headaches were very frequent, but most of them were mild and most of them were infusion related. Last few slides, basically, one of the key lessons we learned from this trial was thromboembolic event. In middle of the trial, we saw a few thromboembolic events. We went to FDA, and then we revised the maximum rate of infusion. So before we revised the maximum rate of infusion, the thromboembolic event were 1.5 per 100 patient months. But after decrease, it reduced to almost 1/3 with 0.5 per 100 patient months. So basically, this trial, IVIG, is the only FDA, EU, and other agencies approved condition for dermatomyositis with efficacy and safety proven in a phase 3 randomized clinical trial. And also, this leads to a pathway, this leads to a precedence and a regulatory pathway for other myositis drug or therapeutic targets or clinical trial to be successful. So this trial, to me, was not so much to prove IVIG, but to at least lay a regulatory pathway to make sure that outcome measure work and prove other things that were very important. And then last but not the least, I want to reassure you that the myositis pipeline is very robust. We have many clinical trials ongoing, phase 2, many phase 3 clinical trials. Obviously, the only successful phase 3 clinical trial is IVIG. We have some negative news for you. Abatacept, which was another phase 3 clinical trial, was recently read out and was a negative trial for dermatomyositis for sure, as well, but in a post-hoc analysis, it showed some benefit in necrotizing myopathy. Linabizumab, which was a trial on dermatomyositis, also turned out to be negative, and a phase 2 trial of KZR61C, which is immunoproteasome inhibitor, was also negative. But in last 1 year, we have 5 new clinical trials starting, at least in my center, and these are all excellent drugs, excellent target. It makes sense. It has a great biological plausibility, and we have a lot of hope from these clinical trials. So to summarize, there are many challenges in myositis clinical trial. We have made significant progress and have internationally accepted classification and outcome criteria. We still need to be developing more objective and meaningful outcome measures for our patients, including PROs. The PRODOM study was the first randomized controlled trial which was proven, leads to a regulatory pathway and precedence or proof that we can develop drugs and get the drugs approved. And also there's increasing interest in myositis with many clinical trials ongoing. With that, I would like to thank you and would open the floor to any questions.
Well, thank you very much, Rohit. That was an excellent presentation. You also served my bad sharing because we started 5 minutes late and we are spot on time. Is there any pressing question from anyone? Well, don't leave yet because Gauthier is still going to do some housekeeping. So thanks again. It was lovely to see you as well in time for the session. Thank you. And these are your Belgian chocolates.
Thank you.
Thank you. Okay.
Thank you to you, Volker, for chairing this session. Thank you for the very, very interesting and high-quality lecture of today. Thank you to the speaker. I have just some housekeeping word for today. We will have now at 11:15 the parallel sessions. Before that, we have the coffee break that will occur in the level -2 in the exhibit hall. I will also remind to you that we will have the e-poster session during lunchtime. Also, I remember we have some remaining possibilities for the networking event that will occur on Friday evening. You can register at the registration desk if not yet done. And this evening we have also the board of the ICNMD for those are a part of this. Thank you to all of you and enjoy the day and the program.